the topic that I was asked to talk about was Mendelian genetics, and most of what you hear about at the Broad in MPG is fairly far from Mendelian genetics until recently, when sequencing has gotten to the point where you can find strong effect genetic mutations directly. But in the past probably 10 years, there's been relatively little done in Mendelian genetics here, because an awful lot of genetic work in that realm was done over the previous 20 years. And so I'm going to try to cover the basic concepts of that and where the modern techniques of genetics lend themselves to really understanding things that were started over that previous 2 decades, but are now moving towards a much better understanding of how you would both understand pathogenesis of disease and attempt to treat it. So we start with extreme basics, which is the human genome, 3 billion bases of DNA. The critical thing to remember about it is it's not just a set of bases on a computer. It's strings of linearly arranged DNA that are packaged as proteins, so that essentially in your chromosomes, where you've got 2 copies of either one, this entire chromosome is a single linear molecule of DNA. Any part of the DNA of that chromosome is attached to every other part, but not attached to any of the others. And that becomes critical for the original approaches to genetics. And when just to be really clear, when I talk about genetics, genetics has really only one meaning. It's not cloning, it's not sequencing, it's not manipulation of DNA, it's not mapping the genome. It's relating differences in DNA sequence difference, or DNA sequence or genotype, between individuals to differences in expressed characteristics or phenotype. Now, expressed characteristics really means anything you can measure. It doesn't refer to gene expression. It's just take an individual, if you can characterize anything, whether it be eye color, hair color, blood group, or level of expression of some small nuclear RNA, whatever you can measure is a phenotype. And if you can relate that, differences in that to differences in DNA sequence, you're doing genetics. Everything else you hear about here is genomics, which is probably a much larger realm of which genetics is a critical component. So this concept of relating genotype to phenotype has been built up with molecular biological techniques over the past 30 years, and the basis for much of the work that my lab started out in the early 1980s, and then which grew into a number of different labs at Mass General, expanded into a multidisciplinary approach, led to the formation of the Center for Human Genetic Research, which a number of the faculty members are also members of the Broad. And we basically follow a particular theme in this center, which is to apply what we refer to as the genetic research cycle, where the view of genetics is that it is a tool. And it's a tool that you can use particularly powerfully because it's unbiased, as opposed to virtually every other tool you use in targeted analysis. This is one that allows you to look at the whole genome at once, and it's unbiased. And as opposed to other tools that you use that aren't directly applied to the genome, this is one that can be applied in an unbiased way, but to a finite universe. That 3 billion bases is finite. You're not looking at an extended infinite universe to try to hope to find something. You're looking, it's essentially, in the early days it was viewed as you had a lake, and rather than fishing in the lake, you weren't really going on a fishing expedition. What you were doing was emptying the water, so that eventually you had to find whatever was there. So we set up to do our research around this cycle, which essentially says, if you're going to study human disease, which is what we were doing at the hospital, and you had patients and families, you could describe phenotype and then apply the rules of genetics to identify the sequences, in this case genes, but we're beyond individual genes at this point, but sequences that are related to that phenotype. Then you get into the realm of traditional biomedical research, modeling in cells, modeling in mice, biochemistry, structural biology, anything that allows you to try and study how that sequence difference caused or is connected to that phenotype. The difference being that instead of trying to reproduce the phenotype, what you try to do is look at the phenotype as guided by the genetics, which is a change in the way that people model, and it's a powerful one, but it's still actually coming to the fore. The goal being to change diagnostics, which is often done very much by just completing this stage of the cycle, changing patient management, having clues to therapy, all of which then finishes the cycle by feeding back here. So I'm going to take you through just some real basics and then an example of a disease that's covered that entire realm of 30 years and where we're at with it and how these various ideas around a cycle apply. So Mendelian disease was in the title. Mendelian disease simply means you have a DNA sequence variation in the genome that is related to a phenotype in a deterministic fashion. Meaning that if you have the variant, you're going to see, and I'm being extreme here, you're going to see the phenotype, okay? So essentially 100% of the risk of the phenotype in a typical Mendelian disorder is attached to a variant at a single location in the genome, and where that location is and how that variant, that functional variant is expressed determines the pattern with which you will see the phenotype travel in a family. So in a dominant disorder where you've got 2 chromosomes, it's called dominant because it only takes 1 copy of the disease gene to pass the disease on to children. So every child has a 50-50 chance of getting it. If they get the functional disease variant, they're going to show symptoms of the disease, and so in a pedigree we'll color them in as red here, and you can track it from generation to generation. A recessive disorder, a single copy is not sufficient. You have to have a copy of the disease mutation or a disease mutation at that location on both chromosomes, and so it takes 2 of them getting together. So if you have carrier parents, 1/4 of the carrier parents, or 1/4 of the kids from the carrier parents on average will show the phenotype. The others, 2 of the others will be carriers, 2 out of 4, 1 out of 4 will be completely free and not be a carrier. Now the difference between these two is fundamental, both in terms of the pattern, but in terms of the likely mechanism of disease. These are typically disorders in which you're inactivating a function. And so you have to completely inactivate both copies in order to get disorder. These are disorders in which you're typically suffering a gain of function or a dysregulation of function where doing it to 1 copy of the gene is sufficient, and the presence of the other copy of the gene is not enough to make up for that and balance the disorder. Then you've got cases where it's on the X chromosome, so it's just a special case of X-linked recessive where in males you only have 1 X, so you don't need to get 2 copies of it to be expressed. But the function, the concept is the same in the sense that loss of function is more common. X-linked dominant, the opposite. 1 copy will work even in females. And mitochondrial, where you're not one of those long strings of DNA, but part of the 16,000 bases of DNA that resides in the mitochondrion, and that travels down through the maternal lineage. So you can look and you can see the pattern of inheritance. The pattern of inheritance says, aha, this is the kind of thing that I'm looking for, but can I figure out where and what it is? The one thing that you hear most about at the Broad is complex disease. Complex disease is simply disease where there is a familial clustering. There's a greater likelihood that you will show the phenotype if you have a relative who shows the phenotype, but you can't discern a simple pattern of inheritance by looking at the pedigrees. And that essentially means that what you've got is a DNA sequence variant that's not a deterministic factor. It doesn't determine the presence of the phenotype. There are other factors that have to be present for you to get the phenotype. So often referred to as multifactorial disorders, where you need many factors to get the phenotype. Okay. So the one thing, when I said I was being extreme because the word penetrance comes in genetics a lot in Mendelian disorders. So if Mendelian disorders were sufficient to always cause symptoms, you wouldn't have to worry about this. But there are circumstances where certain genetic mutations where you can track through families will occasionally be present in individuals who are not at the time you see them showing the phenotype. And that is referred to as non-penetrance at that time and place. So the penetrance is essentially the likelihood that a person with a genetic defect will display the phenotype. For Mendelian disorders, by definition, this is the likelihood that they're going to show it is very high, because if it wasn't very high, you wouldn't be able to discern the pattern of inheritance. But there are exceptions, and I'm going to get into an example of where that's useful. So the fundamental principle, just to reiterate it, is that genetics provides an effective unbiased tool. You don't have to make any guesses about the biology of anything. If you have the phenotype, you can go look for the factors that trigger disease or modify disease, or cause predisposition to disease, or that cause a difference in your reaction to therapy for the disease. Anything you can measure in any way related to the disorder, its treatment, its expression, your recovery, etc., you can use genetics to go after it. So that's why I say genetics is a tool. You could study infectious disease with genetics very easily by determining differences between people in their susceptibility to infection, differences between people in their recovery from infection, etc. Genetics is a fantastic tool because it doesn't limit you into what you think you already know. It allows you to discover new things that you didn't know by being unbiased. There are two approaches that have been used. The one that predominated for many years was an indirect analysis of genotype, which I'm going to go through in more detail, and that is the use of genetic markers. What is coming to the fore now is a direct analysis of genotype, because you can now sequence genomes fairly effectively, and if you can't afford that, you can sequence exomes, whole exomes, and find DNA sequence variants directly. You can, in many cases, although still not in all, go directly to finding a causative mutation and assay the functional variant that causes the disorder directly. In most cases, however, it's easier and has been more applicable over the years because of the absence of next-gen sequencing for most of that time, to use genetic markers to get to the functional variant that causes the disease. And that there are two fundamental approaches that started out with linkage, and it's worth understanding the difference. So linkage studies are cases where you are correlating the inheritance of genotype with phenotype. Okay? So you have an individual, they have a phenotype, you measure their genotype. You're not doing linkage there, you got one individual. What you have to see is that genotype transmitted to the next generation in a manner that's correlated with the phenotype. And that's because of the linear strings of DNA in the chromosomes, which are not in fact passed on intact, but are passed on as linear mosaics by recombination. On average, each arm of the chromosome will have one recombination per meiosis. They're not completely random, but they're also not always occurring in the same place, so they're distributed. And so if you were able to color a chromosome in the grandparental generation, and let's follow this red one since it's got an arrow beside it, that red one, by the time you got to the next generation, might have broken up by recombining with the black one. So you now have a linear mosaic of red and black. And in this generation, that's matched up to a linear mosaic of blue and orange. And so now that undergoes recombination. And so in the grandchild generation, this is what all the chromosomes look like. And if that arrow is a dominant trait, so you have a 50-50 chance of inheriting it from the parent, and it happened that you inherited it here, then in each of these grandchildren, you'd have a 50-50 chance. The ones who got it would have that red region, but might not have any red on the rest of the chromosome. The ones who didn't get it will not show the trait and will not pass it on. That's linkage for a dominant trait, and you're simply correlating the presence or absence of that red region with the presence or absence of the disorder. If the red region is there, the disorder is there, they're linked to each other. This is just an example with a recessive where I told you you needed 2 copies. You can follow it down and see that only the kid that has a green and a blue, because of course it's a different copy in each of these grandparents that is defective, will get the disease. But the point of this is really that if I look and do linkage in a given family with a dominant disorder, I might, in one case, find it tracking with this region that's red. But if a mutation in another family happens to happen on a region that I would color purple rather than red because it was a different mutation, then in that family, a purple chromosome would be linked. So I'd have 2 different families, both with the same disorder, both with the same region of chromosome tracking, but with different genetic variants tracking. Okay? That differs from association, which was, again, you hear a lot more about because of the tools now that make it possible, which is correlating the presence of genotype with phenotype. So in linkage, different families may have different alleles for polymorphic markers near the disease gene. For association, what you're looking at is there a particular allele of the polymorphic marker that's present more often in people with the phenotype than people who don't have it? And in Mendelian disease, you're basically saying, if you wanted to do association, you're saying, can I find a group of individuals who have the phenotype who always have this particular allele? Okay? And that would put you on the chromosome near where the genetic variant was. In the case of the linkage, 1% recombination or 1% chance that 2 spots on the chromosome will have a recombination between them corresponds to about 1 million base pairs. So in most pedigree type studies, you can get linkage to within 5 to 10 million base pairs, and there you can allow for 5 to 10% recombination and still get a statistically significant result of saying I get co-inheritance more frequently than I'd expect by chance. So linkage got you within 5 or 10 million base pairs of a disease gene, and then you needed to go try and find it. Association gets you to a variable distance depending on the frequency of the variation, the frequency of the disease gene, the nature of that disease gene, the heterogeneity of it. There's a lot of factors that come into association, but again, you're going to come to a chromosomal region and have to actually then go look for the actual sequence variant that's causing the phenotype. When you get there, and this is going to be true also of the current mode of let's just go directly in sequence and hope that we can sort out which is the variant that's functional from all the variants that we detect, and in which case both linkage and association can be very helpful in deciding. But when you get there, typically you are surprised. And I put this slide up only to make the point that over the years that linkage and association and ultimately cloning of disease genes in Mendelian disorders was really heavily pursued for a good 20 years. And so that most of the common Mendelian disorders, the genetic defect is now known. In almost all the cases where a gene was cloned, you got a gene that was unexpected, that wasn't one that had been proposed to be involved in causing the phenotype. So we knew a lot less than we thought we knew, because there were a lot of candidate studies that were done where people would say, aha, it's going to involve mitochondrial energy metabolism, so I'm going to go look at these set of mitochondrial genes. Didn't always work out. Works out more frequently now because we know more, but it's still not a great approach. The better approach is to go in an unbiased way and find something new that tells you something new about the biology and doesn't, in fact, give you a bunch of negatives for candidates. Okay, so I'm going to give you an example now of going around this cycle and how this first step, which is what I have kind of been alluding to of taking a Mendelian disorder where you have phenotype within families, applying either linkage or association in conjunction with the phenotype to get to an underlying variant has been the purpose. But in fact, what you really want is the whole cycle. And genetics continues to apply throughout it, and that's really what I want to get across in the end. So Huntington's disease, many will have heard of, is a movement disorder that has its onset typically in midlife, but it can have onset anytime from childhood until you're quite elderly. It's called Huntington's disease because it was described in 1872 by George Huntington, who was a physician on Long Island, a family physician who happened to have a father and grandfather who were both family physicians. So when he was a child, he would go out on their house calls, out in the buggy and drive around the villages and treat people. And he noted when he got out of medical school and started his own practice there, that he was seeing people in the same families that his father and grandfather had seen with this really peculiar movement disorder. So he not only picked up the neurological signs, the critical thing that he did was he also picked up the inheritance pattern. This was pre-rediscovery of Mendel. So he wrote a very nice paper. It was like the only paper that he wrote, essentially, in his lifetime. And clearly it carried a lot of weight. It wasn't in Cell Science or Nature. It was in the Medical and Surgical Reporter of Ohio, that described this peculiar set of symptoms and the fact that if you did not inherit it from your parent, none of your progeny or their descendants would ever have it. Okay? The movement disorder is one that's very insidious in onset. A person is normal to any neurologist, they're normal for most of their life. Then they have this very subtle beginning of adventitious movements. And one of the ways of testing is to have them close their eyes and hold their hands out straight. And what will happen is that an individual who has the beginnings of this disorder, their fingers will actually be going like that. They won't know it. They won't note it at all. The physician will. Eye tracking, the eyes don't go smoothly across. They start to go in a jagged fashion. And that builds to minor movements that lead to clumsiness. So I might, bump into this and drop the pointer and think, oh my God, if I'm at risk for Huntington's, I might have the first signs. They then grow, and an individual gets these kind of dance-like choreic motions so that they are constantly in motion all the time that they're awake. When they sleep, goes away. All the time that they're awake. That progresses over 15 years so that they have to be protected from themselves. You'll see individuals who basically get into positions where they're rubbing their bones against the body or against another limb, and they'll do it so much it'll just wear right through. So they have to pad them to prevent it, or you'll be in a room with a severe Huntington's individual 15 years after onset, and you'll hear a clicking sound, and what that clicking sound is they've rubbed their teeth together for 15 years, and that's the little bits that are left, just going click, 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 because they can't stop the motion. They typically die of heart disease as a result of massive weight loss. Towards the end of the disorder, these people are taking in 10 or 12,000 calories a day, and they're still losing weight. Or aspiration pneumonia, because they get to the point where they can't swallow. And so it goes into their lungs, they get pneumonia. There are also psychiatric symptoms and intellectual decline. The intellectual decline is not as bad as it is in Alzheimer's disease, so that you can still trigger interaction with people with pictures and keyboards that allow them to type, that overcome the movements. So they're really very lucid quite into the disease. And the psychiatric symptoms can sometimes precede the movement, but they're very variable. So some people actually don't get them, and some people get them very severely. There's no treatment for this. I mean, you can treat the symptoms a little bit, but they don't stop the progression. And you die about 15 years after you have onset. Okay, so that gives you what a clinician would think about Huntington's disease. Patient, that's what they'd look for if a patient came in. Yep?
So many diseases there, even the mutation already in the embryonic stage is there. So why they have late onset? So what causes that early onset and late onset?
We're going to get to that. Okay. Good question. The basis for the whole rest of the thing. Okay, now the pathologist's point of view on Huntington's disease is that they look in the brain and they see that this region, known as the caudate nucleus and the adjacent putamen, it's called the striatum, isn't there. It goes away, it dies prematurely. If you looked at an individual before onset of symptoms, by 20 years, they'd look like this. If you looked at an individual at the time that they had symptoms, there have been a few cases where people have died very close to getting symptoms, you've lost maybe 30 to 50% of the neurons in this region, and that progresses over time so that you eventually have lost all of the medium spiny neurons that make up about 80% of that region. There are other cells in the region that survive, but the medium spiny neurons disappear. Now you'll notice the rest of the brain is also shrunken, so there is cell loss in other regions, but it's really the architecture here that's completely destroyed. And while this wasn't known prior to cloning the Huntington's disease gene, there are these aggregated proteinaceous intranuclear and cytoplasmic deposits that can be stained with ubiquitin. These were found actually via a mouse model rather than the human disease. But then when the human brains were looked at, you could find some evidence of these. And that has led over the past 20 years to an enormous industry, in essence, labs. There's probably 200 labs around the world that study this process of protein aggregation in this disease. And there's all kinds of models for various steps that could lead to either the presence of these or their clearance or differences in their clearance and attempts to treat it, etc., that bring in all kinds of mechanisms. And that's what you'll see if you look at Huntington's disease from the pathology and neurobiology kind of approach. Is you'll see a lot of stuff about aggregation and various steps involved in how that can happen. Okay, from genetics, we'll take it back to the very beginning, which is that we did linkage analysis back in the early 80s. It was the first disorder on the autosomes that was mapped purely by linkage to DNA markers. And the DNA markers back then were gel bands that were different in size based on restriction enzyme cuts that either cut or didn't cut at a given sequence because there was a sequence difference, so the site went away. And by measuring the difference in those bands, you could follow the inheritance. And essentially, we had a marker that had 4 different patterns, A, B, C, and D. And you can see in this family that I believe in this case it's the C that is traveling with Huntington's disease. So here's the C going down to here. You got it, you got it. You'll notice in this generation, it's not 50-50 in terms of being black or white, but there are some people with a C. And this is an example where, because the onset is later in life, if you catch somebody who's too young, you can't see the phenotype. So at that age, it's still non-penetrant. Okay? So in our studies, we had to be sure that we dealt with people who were definitively Huntington's disease or definitively free. You couldn't take somebody who was 30 years old and say they were definitively free because you had no way of measuring it. But you could say if they had it, if they were diagnosed. Okay, so we mapped it to chromosome 4. It took 10 years to go from the original linkage to the gene itself, partly because we linked it in '83 and there were no techniques at that point for moving along the chromosome. A lot of the techniques were developed, but the other problem was we were out at the tip of the chromosome. And so any genetic effort attempts to come in from both sides by having markers on one side and having markers on the other side and flanking the gene and going in. Because we were at the tip of the chromosome, we had difficulties in flanking it, which I probably won't go into too much. But anyway, we were able ultimately to flank it to a region of about 2 million base pairs on chromosome 4 with various markers. And again, at that time, we started out with these restriction fragment polymorphism markers. We then moved on to simple sequence repeats, GT dinucleotide repeats, CA dinucleotides that you basically would amplify by PCR and measure the size because, of course, PCR came along during that period and allowed you to do that and discover those. And we ended up with a 2 million base pair region that had no recombinations in families with the disease. So every piece of DNA in that region tracked 100% with the disorder in every family that we could look at, which was a lot of families worldwide because the entire HD community contributed to this. So we then turned to association, which wasn't being done because for Mendelian disorders, linkage got you there most of the time because you weren't at the end of the chromosome, but we had problems. And we turned to association where with these multi-allele markers now, instead of having a chance of having two different alleles at a particular site, which you would now have with a single nucleotide polymorphism, we might have 10 or 12 alleles at a given site because each of these repeats could have multiple different sizes. And so we had great discriminating power to look at haplotypes. And there had been no new mutations to Huntington's disease ever reported because to prove a new mutation, you'd have to find a sporadic case and then prove that the parents didn't have it, which means that the parents would have had to live to a very old age without showing symptoms, and you'd have to get around the fact that about half the people you'd pick up in the first place would be non-paternities. When you're going for something rare, you pick up what is not actually that rare an event. I think non-paternity in the US is somewhere around 10%. So you had a great difficulty just from the nature of the disease proving a new mutation, but there hadn't been any cases proven. So the possibility was that we were dealing with a single historical mutational event that then meant that everybody who had the disease should have the same pattern of alleles around the marker because they would be 100% associated with that particular variant. So we set out to test that with these multi-allele markers and picked up from around the world, I think it was 78 different HD individuals that we typed and phased so that we knew what the haplotype was. And what we found, in fact, was that there were many different variants. There had clearly been multiple mutations causing Huntington's disease, but there were a subset of them that were recognizable as having the same haplotype, where the red here simply indicates that one particular allele at a site that was determined matched. The black are sites that were either indeterminate or that didn't match. I think in this case it was indeterminate in the individual. So that said that at least in that subset, we'd narrowed down from the 2 million to a smaller region. Then we found this other subset that we said, okay, this is either a different mutation but happens to share some alleles with this, or there's an ancestral origin that is the same as this but more distant. And if we take that hypothesis, we narrow down to a much smaller region. So that's actually what finally got to the gene, was this progressive use of association and haplotypes rather than association with individual markers, which is pretty much what was done at that time. And so we ended up finding this gene, and it would have been very hard to find this even now by sequencing because the variant is a polymorphic CAG repeat that expands to cause Huntington's disease, and it expands to the point where it's not effectively captured by today's sequencing techniques. So while we might be able to, and I'll show you we could, get to the region with modern techniques, we wouldn't have just picked up the mutation. We would have had to still do some work and probably some haplotyping to do it. So the mutation is this expanded CAG repeat. Normal individuals have 6 to 34 of these. It's polymorphic in all populations. When it expands to more than 34, you're going to get Huntington's disease if you live a normal lifespan, except for a very small percentage of individuals who have 35 to 39 repeats where there is a degree of non-penetrance. There are people who've died in the 90s with 37, 38 repeats who did not show symptoms of HD. Although on pathological examination there were neurological signs, there were pathological signs. So penetrance in this case depends in part on lifespan, in part on CAG repeat. But once you get over 40, it essentially accounts for all the risk of getting the disease. You can PCR it and size it. Therefore, you can tell in any individual at any time in life, do they carry this disease gene? Are they going to get HD? And this is the distribution in one family of CAG repeats, one very large family of CAG repeats, where every individual has one expanded who's affected and one normal size repeat. So that shows you the polymorphic range of the normals. However, you'll notice that I said one family, and yet not everybody has the same length of repeat. And that's because once it gets into that expanded range, it's unstable. So in every generation, it has a chance of changing size. It typically changes up a little bit or down a little bit with a probably 2/3 chance of going up and 1/3 chance of going down. And then every once in a while, it jumps a lot more, 10 or 20 repeats. And that almost always happens in males. Okay? Becomes important because there were peculiarities in the inheritance of Huntington's disease that were noted. One of the things that was noted very early on is that those cases that had onset in childhood almost always inherited the disease from their father. Didn't matter if they were a boy or a girl, but they almost always inherited it from their father. Okay, so let's say we took today's techniques and went now to look for the Huntington's disease gene, or an equivalent gene, but we'll do Huntington's because we have the data. So if we do SNP association, where we run 1 million SNPs across the genome on one of these arrays, measure the alleles, and say, do Huntington's disease individuals have an association with any SNPs? Are there any alleles that are present more frequently in HD than in normals, a standard case control approach? This is what you get, which is that you get an incredibly high peak right near the tip of chromosome 4 that says, aha, in one experiment, I can take these individuals, I can go run an array, and I can say, aha, the disease gene is there. Now, if I blow up that region, here's what you get. Here's your best SNP, okay? Here's your repeat. The best SNP is sitting in RGS12, which is not the Huntington's disease gene. It's about 350 kb away from where the mutation actually is. So the best SNP doesn't immediately get you to the disease gene. It still, like linkage, just gets you to the region. And in fact, the linkage and association that we ended up with got us to about 150 kb. This association is only getting us to 350 kb, so not quite as good. But it just means we have to work in here. And the reason for this is very simple. The reason for this is, I told you earlier, there's been more than one mutation causing Huntington's disease. This happens to be a relatively rare allele that's on the most frequent chromosome that causes Huntington's disease. But there are a lot of others that don't have it, okay? But over here, these are relatively common alleles that are present on many chromosomes that don't cause Huntington's disease and are present on the major chromosome that does, but the differential's not that great. So it all really depends on the differential when you have this mixture of haplotypes. And if we look at the haplotypes across just the HD gene, this actually doesn't extend out 350 kb. This is just over the HD gene itself, where the repeat, in this case, is right here. So I've flipped it around. This is a set of alleles at a whole series of SNPs that go across that. And the most frequent haplotype is this HAP1 that's most easily detectable because there's an indel in the coding sequence right here. And there are only 2 haplotypes that have that. These 5 and 1, it turns out, are related to each other. All these others are present on different Huntington's disease chromosomes. I've colored this one in green because this one actually is the most frequent haplotype in the normal population. But it is present on some Huntington's disease chromosomes with the mutation. This is the most frequent one in the Huntington's population. So haplotypes become a way of really sorting things out if they're nicely phased and completely determined. And here's the top one that makes up about 50% of the HD population worldwide. And that doesn't mean just the 50% of people that I pick up in Europe and everybody else is different. This is present all around the world. It has spread all around the world. The most recent publication being one out of this isolated population in Peru that turns out it has that haplotype. Here's the presence of that in the normal population. Here's the green one, which is much more frequent in the normal, but still present here. So by actually sorting through these haplotypes, one could home in more effectively on the mutation. So that got us to this point, essentially. We got to the point where we'd identified the gene. We had the ability now to move on, okay? As did the entire community, have the ability to move on, because now we've got the defect, we can start to work our way around the cycle. So the traditional way of doing it that I told you at the bottom of the pathology slide had been going on for about 20 years and had a lot of labs working on it, came from an experiment that starts here. So this is the Huntington protein. So about over 3,150 or so amino acids, and here's the amino terminal region, and these cues are coded by the CAG repeat. So a normal individual will have somewhere 17, 18, 19, 20, 21 cues. And when the CAG expands, this Q stretch goes up to over 37. Okay? Well, the instability of this was something that people wanted to study. So they took a clone that represented just the 5' portion here that contained exon 1 with the CAG repeat, and it turned out by chance that the clone that they pulled, I think it was an EcoR1 fragment, happened to take a piece of promoter, and they injected it into mice because they wanted to get that repeat into mice to study instability, and what they ended up with was a mouse that expressed that fragment of exon 1, but with an expanded repeat. That mouse was incredibly sick, developed aggregates in the nucleus of virtually every cell in the brain, and in fact many peripheral cells, was so sick by 13 weeks that the vets forced the investigators to sacrifice them because they felt it was cruel to keep them alive, and had a variety of tests prior to that that showed abnormalities. There were other mice that were generated at the same time in the same experiment that were not nearly as severe, but this one was particularly severe, and therefore this is the one that people homed in on to work on. And they did two things. They basically said, aha, let's go look in human brain for these kind of aggregates. And while they found some stuff, it didn't look like it did here. And the other thing they said is, this now becomes the model of Huntington's disease. We're gonna cure this mouse, and we'll cure Huntington's disease. Okay? I don't have a slide. One of my colleagues has it. Jung Min Lee has it. It's a great slide that basically takes the literature. It's one of these slides that takes terms from the literature and graphs them based on their frequency, and so you have size and everything. And pre-cloning of the gene, the big terms in the middle were human, genetic, neurological, inheritance, things like that. And after this, the big term in the middle for papers on Huntington's disease was mouse. So essentially, the literature became a study of a mouse disease rather than of the human disease. And I say that because genetics continues. And this didn't match any of the genetics. Okay? This is that blown up model of all the different things, all the pathways that have been studied and various things that have been done. So it's clearly there's very complex set of work that people have homed in from all different directions, and it's been an enormous effort, and it's been terrific for understanding polyglutamine toxicity in cells, etc. But it isn't really what's triggering Huntington's disease. And that's because now that you go around the next part of the cycle, you can do the mouse modeling, you can do the lower organisms, you can do the biochemistry and cell biology, but if you really want to stay relevant to the disease, you better always relate it back to genotype-phenotype relationships, because it's very easy to get off into interesting phenotypes here that don't have any relationship to the human disease and aren't generated by the same mechanism. So now if we go to the geneticist point of view of the Huntington's disease pedigree, rather than just the black or white of affected or not affected that a clinician would be able to tell you, you now can type the CAG repeat, and here you get, for example, a 19 and a 15 in a person who doesn't have HD, and a 42 and a 15 in a person who does, and that person passed it on as a 48 to this child, and as a 55 to this child, and as a 40 to this one, but this one hasn't yet shown symptoms. So that's a typical inheritance pattern for the CAG repeat. But I've also put underneath these the age at which the onset of those motor symptoms occurred, or the age of diagnosis, because diagnosis is based on the characteristic motor symptoms. And so you have here an individual who had 42 CAGs, who had onset of disease at 44. And here you have somebody who had 51 CAGs and an onset at 30. And here's a 41 and a 42, and so on. The one other feature I would show you is, here's a case where there was a mating between two people who had HD. And so that 1/4 of the children, on average, would get both copies. One copy from the mother, one copy from the father of the HD gene. And here's a child who did. Got a 48 and a 42. So they have no normal repeat. They only have the mutant repeat. And here's their onset, 35. Okay? So you could look at that or you could plot it out. And if you plot it out over thousands of HD individuals, the mean age of onset associated with any given CAG repeat is what's plotted here. There's a very strong correlation. The longer the CAG repeat, the earlier your onset of disease. And I will point out that everybody who has Huntington's disease has the same mutation, the same expanded CAG. So there's a very strong correlation. Yeah?
What's the deal with sporadic cases?
There are very, very, very few sporadic cases, and where they have been found now, they have turned out to be expansions from the high normal range of 30 to 33 up into the disease range above 37.
And is the propensity to expand inheritable?
The propensity to expand is?
Is it inheritable?
It is clear that individuals vary dramatically in their propensity to give off altered alleles, particularly males. The degree to which that's a heritable characteristic isn't yet completely clear. It looks like it is, but it hasn't really been effectively measured. Okay? But an individual who has one juvenile onset has a stronger likelihood of having another juvenile onset, but you can actually tell that by just typing the CAG in their sperm, because in that individual, the CAG repeat in their sperm will match the entire world population of HD. It'll do the whole range. So some people are particularly unstable. Okay, but if you now go and you measure the actual individual CAGs versus onset and say, what's the range? You get a range of plus or minus 20 years around that mean. So there's a lot of variation in the onset, okay, beyond what the CAG is causing. The CAG, it turns out, accounts for about, if you take this typical adult range of 40 to 55 repeats or so, it accounts for around 55% of the variation in age of onset. Yep.
Is that related to your ability to measure those clinical phenotypes and that,
you might think that, you might think that, and in fact the neurologists do believe that. They're quite willing to say, oh, it's a hard diagnosis, there's a lot of variability. But the correlations are actually remarkably consistent. We could do this in the US, in Europe, in Africa, where they rarely see this disease, and plus or minus 3-4 years, they're actually really good. I mean, I keep telling them how good they are, and they don't believe it. But statistically speaking, when you put it all together, they're really good. Okay, so here's an important point that I got to finish fast because of the timing, and that is I pointed out there were people who had 2 copies of the HD mutation, and I've plotted them so that their larger copies in red and their smaller copy is in green against all those other people who are in black who only have 1 copy. You'll notice that the reds essentially fall nicely within the distribution of heterozygous. So it only takes 1 copy of the gene to cause the disease. It only takes 1 copy to fully engage it, because the presence of the second copy is not pushing you away to a more severe phenotype. So it's a truly dominant disease. 1 copy, that's it. That's unusual. Most things you'll hear about in humans that are dominant are really not. They're really co-dominant, where 2 copies is worse than 1 copy. Quickly, fundamental principles. Let's see. This is just telling you stuff. Human heterozygous for HTT inactivation have no phenotype, so it's not an inactivating mutation. Homozygosity for those mutations is fully viable, so it's not a loss of function. And it's not, importantly, it's not a dose-dependent mechanism. 2 copies isn't worse than 1. All those animal models that work on aggregation, they're all incredibly dose-dependent. The length of the HTT CAG is the primary determinant of the rate of pathogenesis leading to motor onset. Some of that remaining variation is determined by other unlinked genetic factors, and I say that because the variance that's not explained by the CAG does show heritability, and the normal range CAG repeat isn't impacting on it, but the CAG repeat overall does have biochemical consequences. So it's a functional polymorphism that is altering the activity of the Huntington protein. And the neuronal specificity comes from comparing to other polyglutamine diseases. So that's basically just saying the repeat and the genotype-phenotype relationships give you criteria for understanding what that trigger must be. And that many of the experiments that purport to study HD pathogenesis are not studying the trigger. They may be studying very downstream events that definitely happen, but they're not studying what's driving the disease, because what's driving the disease has to conform with the genetics. And the genetics also gives you an opportunity. Here's a case where you have somebody who's 20 years later onset than expected. Here's 20 years earlier onset than expected. I told you that that difference is highly heritable, so you can go look for the things that cause that in a modifier study. And so what that really means is that if you think about Huntington's disease, yeah, it's a Mendelian disorder. It's inherited 50-50. But if that's not the phenotype you want to study, if the phenotype you want to study is age of onset of Huntington's disease, well, that's complex, because it's caused not just by the CAG, but by other genetic factors in combination. So you've gone from a phenotype where you're dealing with risk of getting the disease, to one where you've split risk and modification. So what we're interested in genetically now, going around the rest of the cycle, is finding genetic variants that modify the disease. They don't contribute at all to the risk, but given the risk, they alter the expression or the timing of expression or pathogenesis. And the reason for finding them, of course, is they may make actually better targets for treatment than the gene itself. And they could act at different stages in life, et cetera, et cetera. And we've gone and we've done, I'm not going to show slides on it, but essentially we've gone and done a GWAS study to try and find association around the genome to sites that associate with altered expression, where our phenotype is simply the age of onset of the individual subtracted from, or subtract, basically the difference between the expected age of onset based on CAG and the observed age of onset. And that has worked in terms of finding locations around the genome that are highly significantly associated. So that by itself demonstrates the heritability of the phenotype, and that there are factors that can impact. Modeling in the mouse has also been done another way, and that is by reproducing the genetics of the disease, placing the CAG repeat into the mouse and having different allele sizes. And by doing that, it essentially eliminates that whole set of work as being involved in triggering pathogenesis, because it now introduces phenotypes that you can measure reproducibly that show a strong relationship. There's no strong, basically they show the same pattern as the human genetics, which is that they are not dose dependent and they are dependent on the length of the CAG, with them occurring progressively earlier in lines that have longer repeats. So you can model this in the mouse, but the mouse doesn't go on and get Huntington's disease. The mouse only lives 2 years. Okay? So you can't expect to reproduce the phenotype in the mouse. What you can reproduce is the genetics, and then you can apply all kinds of tools of molecular biology and everything else to getting to the gene. So the final conclusions are, Mendelian disease is simply a genetic defect that's sufficient to produce an observable phenotype on the common human genetic background. Actual disease manifestations may be modified by other genes or by environmental factors, indicating that the comprehensive phenotype shows complex inheritance as opposed to Mendelian inheritance. Genetic modeling best relies on matching genetics rather than phenotype. I didn't necessarily emphasize this as much as I often do, but this is really, really, really important. You can spend years studying a phenotype that doesn't matter if you don't match the genetics. And ultimately, when you get to things to genetic variants from any of these other approaches that I talked about in complex disease, those individual complex disease variants will have alleles that act in a Mendelian, that they themselves act in a Mendelian manner. They may not have a Mendelian effect when integrated with all the other variants that impact on them, but there should be something about them that you can measure that is having a measurable effect in a dominant or recessive fashion in a bipartite set of alleles. And so that, I believe, is that. Yeah, thank you.